Hello everyone, welcome back to the BioTuber. I am Swastik and today we are going to discuss about nucleic acids and nucleotides. So we will start with the nucleic acids. Now the question comes, what are nucleic acids? Nucleic acids are one of the biomolecules or one of the major family of biomolecules found in the living world or in the living organisms. Now these are the molecules that code for genetic information, and these act as the inheritatory unit for transmission of information from one generation to other. Usually these biomolecules or nucleic acids are present in the form of DNA or RNA, and these, if from a broader point of view, controls the overall machinery of living organism. Now the question comes, how? So DNA, as we know, that can be transcribed into RNA, and the transcribed RNA can be further translated into proteins. And proteins, we know that they control the overall machinery of the living world. So from a broader point of view, the nucleic acids are the major regulatory biomolecules. Now, if we look into the history point of view or historical point of view, the nucleic acids were discovered by Friedrich Mischer in the year 1869. At that point of time, he called these as nuclein because this was the noble molecule that was isolated that time from nucleus, and it did not match any of the known biomolecule at that time. And those were proteins, carbohydrates and lipids. Actually, Friedrich Mischer was working on discarded surgical bandages, and from that he was able to isolate pus cells or leukocytes, and from that he isolated this new biomolecule, which later came to be known as the nucleic acids. Now, why it was called nucleic acid then? There are two reasons of the naming. Because first, it was isolated from the nucleus and thus the word nucleic, and since it has a phosphate group which imparts acidic property to these molecules, these were called as nucleic acids. And after further research and investigation, it was also found that these nucleic acids are not present as such inside the living organism or cells, but are present in the vicinity of basic proteins or wrapped up around basic proteins, which further came to be known as the histones. We will look into those in detail in further classes, and for now we will consider these nucleic acids only. So the nucleic acids are basically polymers of nucleotides. That is, many units of nucleotides are joined together to form the nucleic acids. And as I have mentioned earlier, the nucleic acids are present in the form of either DNA or RNA inside the living organism or inside the... Okay, so the nucleic acids are present in the form of either DNA or RNA inside the living organisms. And these DNA and RNA differ because of the presence of a 5-carbon sugar, which is different in both of them. So let's look into nucleotides and see what are the differences and how these are made. So nucleotides are the monomeric unit or building block of nucleic acids, and these are composed of 3 major groups or families. First, our nitrogenous base. Second, a 5-carbon sugar, and this 5-carbon sugar can be either a ribose sugar or a deoxyribose sugar. And because of these difference, actually the DNA and RNA are different or classified differently. In case of ribose, it is a simple sugar, 5-carbon sugar, but in case of deoxyribose, there is an absence of hydroxyl group in the second position of the carbon, and that's why it is a deoxyribose. And the third group is a phosphate group, which imparts the negative or acidic property to the DNA or RNA. So let's look into the details of each of the component, and we will start with the nitrogenous bases. Okay, so let's take a look into the nitrogenous bases in detail. So the nitrogenous bases are basically nitrogen containing organic molecule, or from, as we can see from the figures, these are aromatic, cyclic or heterocyclic molecules. Now, due to the presence of nitrogen, these are called as nitrogenous compounds, and the bases it is called because of the presence of some basic character, and this basic character is imparted because of the presence of a nitrogen. So if we look into the structure of nitrogen, the nitrogen has 5 electrons in its outermost shell or the valence shell, and 3 of these are covalently bonded to other atoms or other molecules, and these two, or there are 2 electrons which are left unattended, which make a lone pair. And according to the Lewis acid base definition, any electron which is non-bonded can sometimes be donated to electron acceptor, and the compound which will be donating the electron is termed as a Lewis base. So from that point of view, the basic property is present in case of nitrogenous bases, and thus they are called as nitrogenous bases. Now, so the nitrogenous bases are broadly divided into 2 categories, that is the purines and the pyrimidines. So the purines consist of adenine and guanine, whereas the pyrimidines consist of cytosine, thymine and uracil. If we look into the structure, the purines are basically fused heterocyclic rings. Heterocyclic because there are presence of nitrogen and carbon, and heterocyclic by definition means in a cyclic compound, if there are 2 or more different kind of atoms, these are called as heterocyclic. So thus these are heterocyclic and fused because there is a 6 carbon ring and a 5 carbon heterocyclic ring. So that's why these are fused rings in case of purines, whereas in pyrimidines these are single rings are present. Now why these are called purines? Because if we look into the history of purines, this was first experimentally isolated or discovered from urine or uric acid. So that's why pure urine, and from that it came to be known as purines. And in case of adenine, there is an amino group present or attached to the purine ring, whereas in case of guanine, in addition to a amine group or a amino group, a oxygen atom is also attached or double bonded to the purine ring. That's why the adenine is very often called as 6-aminopurine, whereas guanine is called as 2-amino-6-oxypurine. Now the nomenclature is a bit different from the normal nomenclature because the naming of heterocyclic fused compounds and heterocyclic compounds follow the IUPAC naming or IUPAC nomenclature, and that is a bit different, in which the heteroatom needs to be given the lowest possible number. So in this case actually it starts with 1, 2 and 3, so that these 2 nitrogen atoms get the lowest possible value. And in case of pyrimidines, the naming is reversed. Actually it is given 1, 2, 3, so that it can accommodate many other functional groups as well. So the adenine and guanine differ from each other in the presence of an oxygen atom attached to the purine ring. In case of pyrimidine, whereas the cytosine, thymine and uracil, 3 different pyrimidines are present. But out of these 2, cytosine and thymine are found in case of DNA, whereas cytosine and uracil is found in case of RNA. There is not much difference between thymine and uracil. It is only the presence of a methyl group which is there in the 5th position of thymine, whereas it is absent in case of uracil. And if we look into the structure, in case of cytosine there is an additional amino group and an oxygen group, whereas in case of thymine there is a dioxygen group and in addition to the methyl group. And in case of uracil there is 2 different oxygen groups which are present at 2 and 4 position. So now that we have seen the different types of purines and pyrimidines, we will look into how actually these purines and pyrimidines are arranged or present in DNA and RNA, and how they are associated with the ribose and phosphate together to make the nucleotides in the DNA and RNA. And it is interesting how actually these 5 molecules are chosen, or why these nitrogenous bases are chosen to be taking part in such a big process, that is the DNA and RNA, which hold the genetic information. And that is still an unexplored area, because out of all the various molecules, why only these molecules were chosen? So that is something we need to consider, and we will look into how actually these are present, or how these molecules are arranged in the DNA and RNA in the subsequent classes. So that will be all for today. If you have any queries regarding this topic, you can put your query in the comment section and I will definitely try to answer those questions. And if you like this video, do subscribe to my channel, like, share, and also click on the notification button so that you will be notified when I will be uploading new videos. Thank you.